part of what I had a tour through Australia planned. We had 10 3-day events that ended up being cancelled. I was in Kangaroo Island doing a fire recovery workshop, soil health workshop, when everything started to shut down with COVID. So my book tour for the love of soil ended up not being, just didn't really come to a lot in terms of Australia and New Zealand. So when I could see the situation developing, especially in New Zealand, I ended up catching a flight. I caught 6 flights and ended up here in Montana, which is where my horse trailer is. So currently you are sitting in my boudoir, living room and kitchen all at once, and I travel from ranch to ranch in North America, which I just absolutely love. So for those of you that haven't read the book, I go into some of the stories of working with ranchers here, and I'll cover a little bit of that as we go. So feel free to pipe up. I might have to get you, Sarah, to ask me the questions because I can't see the screen right now. So there were a couple of questions just thinking about weeds. It's something that we seem to get quite fixated on, and something that I get really excited about because they are passing so much information for us if we learn to really key in and listen. So what is this particular species trying to indicate? And so there's 6 main reasons, and those reasons related as to why a plant will germinate. So firstly, to colonize bare soil. And there's probably quite a few of weeds that I think immediately will come to mind for that, or in response to low organic matter. I'm not looking at any of you on your sands. Sorry, silver loams, red loams. Maybe to open up compacted soils, or in response to mineral availability. Perhaps there's a microbial imbalance, or as a safety valve for toxins. So in the book I go fully into quite in depth into these different aspects. So one of the aspects might be, who's that? I can hear something really crackling in my ear. I don't know if someone still needs to be muted. So yeah, one of the things that we see that can stimulate different types of plant species are what we call disturbance events. So these disturbance events could be natural or unnatural. So it could be fire, it could be flooding, could be extreme winds, or it could be things like cultivation, synthetic fertilizers. That photo down the bottom, that's a DC3 applying superphosphate. So that's what my grandfather used to do. And what was interesting is one of the things they used to put into superphosphate in the 1960s was DDT. So DDT had an impact in terms of microbial function and microbiology. So I want you to think of weeds in terms of disturbance events, right? So I have this disturbance timeline. So some of you will be familiar with this in terms of the soil food web. So thinking about soils that go from very primitive conditions, from bare soil, and that could be your cryptogram environments, to soils that don't have a lot of disturbance at all. So our deciduous or our conifer forests, where we see a whole lot of fungi growing in that environment, to soils that are highly disturbed, which have a lot of bacteria. So around the central part is often where we're aiming for in terms of what are the species that you're trying to grow. Are you trying to grow perennial grasses? And around that zone is where the bacteria to fungi ratio is 1:1. So where it says 1:1 there is actually off. That is actually 1:4. So I want you to think of grazing in terms of, is it a disturbance? And what you find is that grazing can shift to high disturbance or low disturbance, and especially if you have very, very large pasture sizes, which is common in semi-arid, arid environments, is that, you know, we just have these massive big pastures that animals will basically meander through, and we end up with this mosaic landscape where we'll have some of what we call the woody weeds, and some of the cryptograms can be in that same environment, because animals will be over impacting what they really love and then under impacting what they just are going to ignore. Like the ice cream is not there, so they're just not going to spend time there. It might be it's too far from water sources or whatever. So this disturbance, you know, manure and urine can really wake up a system. If we don't have any livestock, then we can see systems start to deteriorate because you haven't got biological stimulation in that system. So it can we can actually see those environments as totally shut down in terms of activity. All right, so some of you will be familiar with some of these images. These are our cryptograms. So what they're doing is they're actually providing protection to in situations where we might have bare soil. And I think historically they've got a pretty bad rap. I know if you look at some of the different grazing management, they will certainly talk about, you know, how to get rid of them, but they are actually doing some pretty amazing stuff. So they're significant sinks for global carbon. So anywhere between 2 to 7.4 billion tons per year, which is about 7% of the entire carbon cycle from land plants. So 7% comes from cryptograms. In nutrient-limited environments, they may be providing nitrogen up to 100 kilograms per hectare per year, which is nearly half of the global nitrogen cycle. And if they are rough, like they got that rough, so cryptograms are groups of like our bryophytes, our cyanoorganisms, so cyanolichens, cyanobacteria. They're basically just different types of very primitive colonizers. And if they have a rough surface, they can help water infiltration, but when they're very smooth, they can actually slow or stop infiltration. So they're not all bad, but it's kind of like if you're going to be impacting on them, then you need to be making sure that your management's creating grasslands or whatever you're after. So I just want to share the story of Betsy Ross from Betsy Ross Grass Fed in Texas. If you have not come across Betsy Ross, you need to get on the Google and check her out. She's in her early 80s, and man, this lady is a hoot. I went and spent a really fun few days with her, and one thing that she was doing was some trials on mesquite. So mesquite, for those of you not familiar, it's created quite a lot of invasiveness. Well, people have created invasiveness in Texas, has very large thorns on it, can turn into quite a large tree. And what she did is she tested the leaves of the mesquite to look at minerals and mineral balances. And then what she did is she applied a foliar, and what the test showed the mesquite was high in. So if you take this test, and you could do this if you're seeing some kind of woody species that are encroaching, so I don't know if it's a mulga or an acacia. So mesquite is a nitrogen fixer as well, so very similar to what the acacia is doing. So she tested it for minerals, and she found they were very high in some specific trace elements. And so what she did was she made up a brew, and she sprayed what that test said it was high in. So let's say copper. She sprayed a little bit of those elements back onto the mesquite. And what was interesting was within weeks, the cambium tissue started to crack. And so that's what the mesquite looks like now. And so these plants all started, within weeks it was cracking, but you know, it's taking a couple of years for these trees to totally die. But what's interesting is like all the thorns fell off them, animals started to browse on them, they no longer were something that was a problem as such. So she just got curious about what is the mesquite trying to do, right? So it's nitrogen fixing, and it's bringing up minerals. And by putting those minerals back on, the plant did not like it, all right? You're doing that plant out of a job. This here is the Chihuahua Desert. It's the largest desert in North America. Average rainfall about 6 to 7 inches. Yeah, just south of the Mexican border, right? So very hot, very dry, very high pan evaporation. And I had the pleasure to meet Alejandro Carrillo, and I'm going to go and hang out with him in November, and I'm very excited about that. But what he's doing is using animal impact to restore these arid landscapes. So from the photo on the left to the photo on the right. And often it's really hard to look at, you know, images like this because you need to see them at the same time of year. You know, like you go, oh, well, that's all very well, that's summer or whatever. But typically his neighbors have huge amounts of open ground. They have a lot of mesquite, and just through increasing water systems, so he's put in some more central water and using basically using what we call range riding or in-herding or migratory systems, mobbing livestock up, putting all your mobs in together and using them as a tool to bring the grasslands back, basically. So this is a good fence line image of what's happening at his place. You'll see this on the Savory Institute website. But how is it that you can actually use animals as tools to get these systems starting to restore themselves? So bringing carbon back, stimulating microbiology. And so, for him, there's no such thing as a weed if we're thinking about weeds. Another example of working, like I talked about, mineral imbalances or microbial imbalances. In a lot of these semi-arid environments, we have a lot of microbial imbalances, right? So when systems get really dry, biology just turn what I call sleepy, right? Not a lot of activity. So Steve Charter here at the Too Lazy 2 Ranch, so funny. He lives about 30 minutes north of Billings, so sort of central western Montana. That photograph on the right was taken when we'd had one of the wettest springs on record, which has been great. But he's actually started worm farms in the middle of his fields. So anywhere he's got piles of like old hay and manure and any kind of buildup, he's been throwing worms at them, and not too concerned if they were going to survive or not, because this place, I think it's an average of 9 inches of rainfall a year, and really high winds, as he wasn't too worried if the worms are going to survive or not. So on the left, we're actually building worm farms and getting worms going, and building those worm systems basically with waste that comes out of his yards, where he might be bringing in some cattle or he's got horses, and then just using that to feed worms. All right. Then what he's doing is he's putting this into a slurry sprayer. So he's got this mounted on the back of an army truck, and he's treating about 2,500 acres a year with a Boomercast extract, basically. So it goes on at about 2 kilos a hectare or 2 pounds an acre. And when this first, we did some trials, he did 400 acres of different types of trials, looking at different types of biostimulants. What was fascinating was just to see the dung beetles fly in. So in his entire lifetime, he'd never seen dung beetles on the ranch. And within just a few weeks of these applications, dung beetles arrived. And it was quite freaky, because I was in a trench. We were doing a workshop, and there was a cow pat in the bottom of the trench, and I'd got a shovel to just kind of throw it over my shoulder so it wasn't sitting in the middle of what we were looking at. And suddenly the air just came alive, like it was buzzing, and I thought it was a wasp swarm or something. Like, it was super loud and scary, and there were just all these bugs flying in, and then we realized they were dung beetles, and they just responded immediately to that scent of this quite nasty piece of dung. So he's got some really tough country, as well as some of this lower country that's easier to get over. And by treating about 2,500 acres, he's able to get round as much of his land as he can. I can't remember exactly how many acres he's got, because he's got two big blocks. Anyway, he's got a lot of land. But what's been really interesting is by putting on small amounts of this worm extract, we're actually switching on the germination signal for a lot of grasses that haven't been very prevalent. He's got a lot of what they call the tame grasses that were planted by, they were advised to plant in the last 30-40 years, that have become quite invasive. They're not very palatable later in the season, and he's been really wanting to get rid of some of those grasses and get in some of the native grasses. And what we've seen through doing this and just changing the signal is that these grasses are coming back, that the seed bank is just sitting in there. So yeah, this is during spring when the situation is looking pretty good. But what he's seen is just a prevalence of very, very palatable grasses, and the livestock just look absolutely fantastic. So his cowboy that has been working with him is just like, I've never seen grass like this. So it's given Steve a lot of confidence to be out and putting more of these biostimulants on as he's able to. So thinking about other ways that weeds are indicating things, many weed species are indicating low available calcium and low humus, organic matter. Thinking in your soils, this low available calcium is actually due to low fungal activity, right? So you have calcium sitting in there, it's just not functional, right? And we'll see some of you probably have very acid soils. Part of that is driven by this microbial interaction, and people might be putting on like lime and keep putting on lime and find that your pH just, it'll come up temporarily and then go back down. We're not seeing calcium come through the plant, because what mediates calcium to the plant is your fungi. So many of your primitive grasses, so coarse grasses, so think grasses that might cut the side of your hands, they're grasses that animals don't perform very well on. I don't know what the native grass species are in your area. Things like button grass or kerosene grass, or someone told me it was called crack grass because or windmill grass, the one that crawls up the back of your trousers and comes out your crack. I love Aussies. You guys are so funny. Anyway, those grasses are potentially, what they're telling you is that calcium is not very functional. Some of those grasses are telling you you've got nitrates or that there's compaction. So learning to read what these different weed species are telling you. Many of our broadleaf weeds, they prefer low phosphorus, not looking at anybody in Western Australia, or very high potassium. So taking these tissue tests, I really, really value it. I get a lot of information from it. So this was a project that we did in the Tom Minor Basin. Look at it. It's just absolutely stunning. Now, these guys have some of the highest predator numbers in the American states. They have 20 permanent bears that are on the property and up to 41 bears during the middle of the season. And part of the issue with these are all grizzly bears, right? So you don't want to see a lot of human interaction with these guys. They're not scared of humans. Is they're coming in to harvest a plant called caraway. So caraway, we did some testing on it, and we looked at what was happening in the soil and what was happening in the leaf tissue, and what we found was that the caraway really liked soils that were very low in boron, very low in phosphorus, and that were very bacterial dominated. And we didn't find the caraway growing where the phosphorus was higher and where we had more fungal dominance. And so just because a plant is there, or the soil has been disturbed or whatever, because the bears are actually digging it up, because it looks like a white carrot. I mean, it's very nutritious at certain times of the year, but they're encroaching closer and closer to the household. So they didn't want to shoot the bears. They're just interested in how do you reduce having them come close to the houses, and what kind of things could we do. So we're putting on biological stimulants and some trace elements to actually change that germination signal, so that we're not seeing as much of these plant species around the houses. Right. So capeweed, and I'm sure no one on this talk has got capeweed, but it's a Cape daisy. It's very invasive. We're seeing it coming along the eastern and western coasts of Australia. Australia, North America. I've seen it in New Zealand now. Take this sample and have a look. Where is it significantly different to a grass species that you're trying to grow? So we compared it from ryegrass to capeweed. So the nitrogen, the phosphorus, potassium, sulfur, carbon were all pretty similar. Calcium, however, was 3 times as high in the capeweed. Magnesium, again, nearly half as high. The sodium was 10 times as high. Zinc, twice as high, and boron 10 times as high as the ryegrass. So that capeweed is a dynamic accumulator, all right? It's bringing up nutrients. It's dealing with an imbalance in terms of functional nutrients in the soil and balancing that soil out. Also notice it's over twice as high in nitrates, right? So we know that capeweed is a nitrate accumulator. It's trying to correct an imbalance in the soil. Certain times of the year, sure, it's going to be protein. Other times of the year, you're risking abortions, you're risking animal health by feeding it on it. And also, if you're feeding animals on high nitrate feed and then slaughtering them, you're actually growing a carcinogenic food. So we should stop doing that. That would be good. So this program we actually put together, and we put these minerals and we put in a little bit of humate to help buffer that nitrate, to help reduce that capeweed, and it's been very successful. So I had some questions about how do we reduce and buffer those herbicides. I feel like we're very much talking about weeds right now, but anyway, there is time for some questions. So you can reduce your herbicide and pesticide use just by looking at decreasing the pH. So you can use a citric acid or a fulvic acid, but just buffering the pH. So get yourself a pH meter if you are using herbicides. I really like using fulvic acid with glyphosate. So you can cut your glyphosate by 30% by adding 1 part fulvic to 4 parts herbicide, and you get the same kill, if not better. So I actually say drop your herbicide. And the reason for that is what it does is, because fulvic acid, so it's a type of soft brown coal that's chemically extracted, so it's a concentrated carbon. When you put that onto a plant, the plant recognizes it as food and it opens up its cells. So you could do that with herbicide, or you could do it with your trace element applications, all right? And we can increase nutrient uptake into the plant, all right? So the plant's like, oh, yummy for me. So yeah, I really recommend, if you are doing this, then reduce your herbicides. Often we are seeing low functional calcium, so let's be lifting your fungal diversity in the system. So there's things we can feed. It might come down to your grazing management. If you're in cropping, there's a big diverse group on this course, so it's really hard to just talk about grazing, but certainly, you know, in cropping, how do we feed our fungi? You know, are we using carbon buffers with our fertilizers and with your herbicides? Everything. Anytime you're out, don't waste a pass. Think, how do I get some carbon out there? I'm going to ignore that middle bit. And then, yeah, address which of the 6 driving factors is causing weeds to germinate. All right, what role do you have in that? I really like this example from the Haggertys, and hopefully some of you have been out there to have a look at what they're doing. So what they notice, they're taking on land that has huge amounts of herbicide resistance. So some of those farmers were putting on probably chemicals. I'm not going to say that they were banned, but potentially that might have been what was happening. Trying to just throw the bathtub at these different types of weeds and not have any success. So these guys do use a buffered herbicide. They put it on with a worm extract, and then down the drill they're putting 5 liters of worm extract, and then they're doing 100 liters of a compost extract when you have canopy closure. In the first year, what they noticed over summer growing was your kerosene grasses, your button grass, your windmill grasses. And then in the second year, they saw this coming up along the drill rows. So this is called Paspaldidium or Serratia grasses, right? And this happened across thousands of acres on their property, but it didn't happen on the neighbors. So it's not just about soil disturbance. It's about, there's a germination signal being sent to these advanced grasses to germinate, right? And now you've got ground cover, and now you've got feed for sheep, right? So yeah, I highly recommend that get in touch with the Haggertys and go and have a look. Now this is, I mean, that was exciting, but this is even more exciting, right? Is they sent in samples. They had 2 lots of seed samples, or soil samples with seed in it, one from the area that was being treated, one from the area that wasn't being treated with the compost extracts. And what they found was, so the glyphosate prospect and trifurulin prospect, that's where it's had the worm extract, is what you see is that those weeds have been controlled at half the rate of chemical that they would normally, that, you know, the label recommendation rate, whereas just outside in areas that hadn't been receiving the compost extract, those still have very high herbicide resistance, right? That herbicide resistance that we're seeing around the world is not due to a herbicide. I mean, herbicides are causing part of the problem, but it's a microbial issue. How do we support beneficial microbes in the system and have systems that are healthy? Because we find as we drive these systems more and more bacterial, it gets harder and harder to kill weeds. So we are seeing over 300 different species now in the world that are herbicide resistant, and it's getting worse and worse and worse. And we could, you know, put dicamba with 2,4-D, or we could look at what are the drivers, how do we get biology to actually work for us and in the system. So yeah, that was sorry, that took longer than I thought it was going to, but that was in response to the questions that came through. So I'd love to hear from you. I know, Sarah, if you've got questions that have come through, because I can't see anything.
Okay. Yeah, no, we've got a couple that have come through. The first one was with the guy in the desert. Are his neighbours set stocking?
His neighbours set stocking. Yeah, they just allow cattle to wander around. Yeah, but very large
Cool.
properties, right? Very large properties. So they're not, I mean, you're talking, 10,000, 20,000 acres. So yes, set stogging.
Yeah. Okay. And the other one that we had was, what is the optimal timing for Vermicast application?
Yeah, it's quite interesting. I mean, I used to always advocate that we be using worm extracts when plants are actively growing. So let's say early spring, or in you guys' case, in winter. But what we're finding is we are getting responses even in the middle of summer when things are really dry. And the reason for that is we're not putting on living biology as such. We're putting on the quorum signals. So these are proteins, metabolites, enzymes that the microbes respond to, and they are more resistant to breakdown than trying to just spray living biology on in the middle of summer. So, I mean, I think you're better to put things on with moisture or to put things on when plants are actively growing. But I also recognize that we are limited in a lot of these environments, and sometimes you just got to do what you got to do.
Cool. Okay. Where there is another one. Is compost tea as effective as compost extract?
It's a good question, and I think compost extract and compost teas are quite different. So compost teas, you are brewing them, you're feeding it, you've got to put them on very, very quickly. They'll go anaerobic. Whereas a compost extract, you're just running water through it. They will actually store very well. We use more compost. So as to efficacy, you can really concentrate a lot of bacteria in a compost tea. I don't think you get as much diversity as you do in an extract, but with more bacteria, you can get better leaf stick. I used to do compost teas commercially, and now all I do is extracts or slurries. So slurries, we're putting a lot more compost material on, but I just kind of think I'm sending the microbes out with their home, because we are putting more compost or whatever out. So yeah, I think you've just got to look at what your situation is and how often you could put something out. In a horticultural system, sure, we could do more compost teas. It's easy, but I think on large scale or broad acre, it's not practical to do compost teas. We're going to do extracts and slurries. Yeah.
Cool. Okay, so we've got quite a few questions coming through, so I'll run through them. Okay. Oh, that's that one. Oh, it's jumped around. Someone's added something. Okay. Can you recommend any good publications on weeds as soil health indicators?
You could read my book. It's got a pretty good comprehensive chapter. But another good book would be When Weeds Talk by Jay McCaman. McCaman, McCaman, and you can. It's an American book. So it's not going to cover all of the. Australia has some really gnarly weeds. So I think we're building more of a library in what we're seeing and through some of these mineral testing and soil tests. So yeah, I do recommend you go and do a soil mineral test, a soil microbial test, and leaf tissue test and figure it out for your own environment. But that When Weeds Talk would be a good start.
Cool. I'm just trying to. Martin's asked a question here. Just trying to work out. Yeah.
Themes.
You have half answered it. Okay. How much research has been done on specific weeds in relation to soil issues? In some areas, we've got a problem with datura. Is it?
Datura.
And fireweed.
Yeah.
Sorry.
That's all right. Datura. Yeah, I just did a presentation in Queensland, and I came across some work on datura. There's a trace element that's involved. I can't recall it. Sorry. But I'll pull it up. So when I send through this presentation, I'll add what the datura indicators were that we found. A lot of research and I'm not to beat up research, but a lot of research is not really focused on what are the causal factors or the driving factors. So we're not really seeing this work being done. It's certainly being done on farms with farmers themselves, as opposed to research work that's happening in it, unfortunately, especially because there's not really a product. It's going to be, you know, how does your management influence these weeds? Because the next question is asking about African lovegrass and serrated tussocks that thrive in lower nutrient environments. I think, Jono, if you have a look, it's not just a lower nutrient environment, but there's a biological component. And if you dig up a lot of those species, they have very shallow rooting systems. They are, when I was talking about your bacterial grasses, African lovegrass and the serrated tussock both fall into that. So environments that are very bacterial dominated, they are being generally overgrazed or not managed as well as they could do. I'm just trying to think. We've had some good results with needlegrass, and certainly some of the other tussocks, but I haven't got any clients right now that I'm working on with African lovegrass. Samantha, the amount of water to compost you said to create a slurry, I didn't. That's going to depend on the type of equipment that you've got. You want to be able to have a sprayer that's, you know, and when I'm talking about slurries, they have very large nozzle sizes, so you're generally putting a lot of water on, like maybe like 500 to 1,000 litres a hectare, because so much stuff is going out with the slurries. So that could be a limitation in some of your environments. But yeah, so it's going to depend on the equipment and your nozzle size. Sarah, did you have something that you can see?
Oh, yeah, there's a whole lot more. I don't know how many more you can see, but yeah, they keep going.
Okay, all right. So with the desert trials, how can we improve our ground cover and native grasses in northwest WA on Pindon with predominant spinifex country, especially when we have over 1 million acres, efficiently and effectively? Thanks. Yeah, Haley. Yeah, not easy stuff. But this is where, you know, we really look at how do we improve what we can, and look at improving close, you know, like areas. Like if you look at the example with Alejandro, he has improved probably 50% of his ground, because how feasible is it to do all of it? And, you know, this is where it comes down to having water sources, being able to move cattle so that you don't have them just set stocked or wandering wherever they want, just having a bit more management on them. But yeah, you know, I don't know what your cattle situation is, but you could be running wild Brahman for all I know. I mean, they could be quite unmanageable. But yeah, so we're doing things like putting seeds in the mineral so that we are using livestock to help germinate diversity of species, using salt and mineral blocks to try and encourage them to maybe beat up some areas. But yeah, I think you just got to, it's like how you're going to eat an elephant. You're just going to take one bite at a time and just start improving where you can, and as you go. But yeah, 1 million acres is a big ask. But yeah, I think the virtual grazing is pretty exciting, and I think that's probably, for me, one of the few tools that we're going to have, is being able to manage them using technology, really, so that you can get control on their movement. When there's not a lot of feed, you really need to be making sure that you're looking after animal welfare. I'm getting quite frustrated seeing people just kind of like, oh, I'm gonna, you know, hit this ground really hard, and not have a consideration for the fact that animals haven't got enough to eat. Yeah. So in a large-scale grazing operation, low rainfall, how should bare soil be approached? Through that same thing, David, as I was saying with Haley, like, You want to assess why is it that you've got bare soil. You know, this idea of holistic grazing where you're trampling organic material, it's not going to work if you've got bare soil. Is it feasible to roll out bales or get some kind of carbon materials? You know, if you're on 1 million acres, probably not. But yeah, I mean, it's tricky because we are seeing the degradation of landscapes across the whole planet, and it's spreading fast, and we really need to get proactive and go, well, is it always low rainfall? Do you ever, you know, like every 4 to 7 years, maybe do you have some kind of monsoonal effect where you could actually be trampling some kind of material? But yeah, livestock really are the solution to it. If you've got bare soil, how big is that area? Could you night corral animals so that you can just dump a whole lot of manure and urine in there and, you know, have seed in your mineral box and just try and get that stimulant going? So a question, how do you suggest people test their compost for microbial activity? If you haven't done a microscope course, then send it off to the Soil Food Web Lab or Microbe Labs in Adelaide. You need to be testing your compost. I wouldn't be using compost and I wouldn't be selling compost unless I had a test. But if you're able to use a microscope, then by all means, just start looking. You don't have to be proficient in a microscope to be able to go, wow, actually that's a nematode, and you know that definitely looks like fungi, and you know, some of the higher organisms that would tell us that compost is in good shape, like a lot of our protozoa and our nematodes. If you're seeing that, they often can be a really good sign, unless of course it's lots of ciliates. So doing a microscope course, I think, for anyone that's making compost is a good idea. And there's lots, obviously now that we're all on lockdown, there's a lot of online courses. So yeah, I really recommend that. The question is, what's the best time to apply whitewash or the fish to cereal crops on ground prior to seeding, or just post seeding, or as a foliar? Potentially for that as a foliar. I want to get it on actually the leaf surface to help with mobility. That fish is going to give you it's going to give you a whole lot of amino acids, so we're getting a bit of a nitrogen hit. What conditions are most favorable? Ideally, you know, in an ideal world, you know, early morning, late afternoon. You know, if it's going to be really hot day, I'm probably not going to go out in the middle of the day. What you can see sometimes is a little bit of leaf burn, but it's all right. The plants will recover. Yeah, so that would be fine. Someone's asking, how sensitive are cryptograms to rangeland grazing strategies? They are very sensitive to them. There has been studies on cryptogram recovery after they've been eradicated. Yeah, there are studies online to have a look at that. But yeah, it comes to sensitive grazing management. Yeah, and for the guy that's got the bare soil, if you've got cryptograms on it, maybe just leave it. Like if you have got a lot of bare soil. How do you suggest getting fungi into acid soils? They are there, they're just not active. So it's about switching them on. So can we do that with our grazing management? Absolutely. If we're trampling organic materials, my favorites really are the humic acids and fulvic acids to really just get fungi very excited. So humic are part of that, our complex foods, the complex carbons, so soft brown coals again. I see more and more people in Australia now are using biochar. That's something that can be very helpful. Just look at where the source of the biochar is. I'm seeing some really dodgy biochar out there, like people making it from tires. So biochar is made in pyrolysis machines, so without the presence of oxygen, they're burning carbon-based materials. They're not making any extra carbon, they're just consolidating what they have. So biochar could work in that situation too. But yeah, think complex foods. Yeah, get them woken up, get your management spot on, and stop killing them. So, you know, if there's something that you're doing that you know might be taking out your fungi, maybe stop doing that. So, you know, fungicides, pesticides, soluble phosphate. can all reduce the amount of fungi that we have in your system. What are my thoughts on saline land? Is that a good place to start implementing practice changes? Or more broadly, is it a good idea to focus on poor performing areas first? That's a really good question. Lizzie, I would recommend you focus on increasing your higher performing areas first, just because there's a bit of a lag and a drag on those lower performing areas. You're actually better to focus on where systems are working well and then pay it forward, because like some of my guys, we're running probably 150% more livestock on those same areas that are performing well, and then use that to fund then improving some of that really low quality stuff. They're predicting, I think they're up to 50 million acres now in Western Australia that has gone alkalized, saline, sodic. It's all about land management, about putting the trees back if you can, and building humus, building organic matter. So humus, organic matter, those humates are all the antidote to saline conditions. The Haggertys that I gave as an example are seeing their saline patches shrinking. So that's just through 5 litres of a worm extract, 100 litres of compost extract. You could be doing that all yourself, very, very low cost, and actually shrink your sodic. We're also using gypsum on some of the saline land with great success. So don't give up on it, but I would recommend you focus on your better performing areas first. Someone's saying there's a farmer working with managed stock and camels on the edge of the desert. Oh, that sounds cool, Samantha. I'd like to see that. With tree browse over 250,000 hectares with amazing results. Camel better with water, but the browse is important. Yes, the browse is important. So the browse isn't stopping the grasses, people. So we you still want to see that shrubs system, but we don't want to see, sorry, itchy nose out in the dust today. We don't want to see that encroachment where suddenly livestock can't get in and we get these areas that just exclude them. But we do want to see browse, we do want to see tree species in there. And, in a healthy ecosystem, the grass, especially in these areas, often they were open savannas. There was, grass underneath those trees. Question, out here in the, you're out here in the rangelands, Jardine. I've heard you mention providing cattle or other stock with good bacteria through watering points to help put more biology through manure. Any other recommendations on this alongside rotational rest-based grazing management? Yeah, so probiotics in the troughs are fantastic. I know some people are putting like Boomercast in their troughs or compost. I wouldn't do compost or Boomercast for longer than 3 days. There are products like effective microorganisms or the lactobacillus recipe that's on my website under resources that you can make yourself and then put out through troughs. And what we see is the really rapid breakdown of that manure, which is fantastic, and we're starting to get that nutrient cycling. The other thing that's super fun to do is actually feed cattle humates. And you can mix that humate with molasses if you want to. So the humate, this is the soft brown coals. What it does is it relaxes the villi of the gut. It aids in nutrient uptake. Anytime you're seeing animals under pressure, so it might be like you've had a diet change and maybe they're scouring, or there's been too much protein, or maybe you've got some kind of mycotoxin or alkaloid out there, which I imagine you guys have a bit of, I'm guessing. But so the humate will actually bind to any of those toxins. So I had a case with a rancher that had ergot. So ergot is it produces a mycotoxin. It's a fungal fruiting body. You'll see, like you might see a ryegrass or whatever, and it's got all its seed heads, and then one of those seed heads will be twice the length and it'll be black. They actually use ergot to make LSD. If you wanted another enterprise, anybody. It's been linked to what caused the Salem witch hunt. So people were actually hallucinating, and it was in the grain. But what it does is it restricts blood flow to the extremities. So for this ranch, hooves were falling off, ears were falling off, tails were falling off, like really, really major. We put out humate, and within 6 hours that ergot was out of the bloodstream. So being able to have that humate out as a free choice, I just think is gold. Like, you know, you're crazy kind of not to, because it is going to really help when maybe forage quality isn't the best. Maybe you've got moldy hay. I don't know what you're doing, but yeah, you'll really like it. So I'd put some humate out there. So what are you saying, Samantha? The Haggertys grow the hay and put it gradually over the bare soil with rounds of their sheep and take them off as they lose condition, rotating the animals. I'm not sure what you're saying. Yeah, I mean, you can drop bales if you do have bare soil, and drop bales on saline areas if you can afford to do that, and graze livestock. David's saying, what can you apply to make calcium more plant available? So yeah, anything that's going to feed you fungi is going to help with calcium mobility. I really do like CAN, so calcium or calcium nitrate, but it's expensive, so probably not that applicable. But the liquid, like the old whitewash recipe, is really going to help, and we're putting that on at like 15 or 10 to 20 kilos per hectare, or 10 to 20 pounds an acre of lime. So it's just the old whitewash recipe. So it's just hydrated lime, burnt lime, slaked lime, whatever you call it, and then you add it to water, mix it up, and then apply that. That's going to really get your calcium mobilized, and again with some kind of carbon source, so a fish or a seaweed or a humate or anything that, yeah, that's going to give them a bit of carbon. Okay, question: is it okay to mix a small amount of UAN to whitewash fish as a cereal foliar at the 3 to 4 leaf? If not, when would be the optimum time? Yep, you could do that, but when you're talking small amounts, like we mean small amounts, like probably 5 kilos, right? Just, you're not going to need a lot. It's going to help. And again, yeah, with the fish, that's really going to get it going. We have seen a little bit of burning with UAN, so I think, yeah, I think that the fish is going to help it, and just make sure you're diluting it. As a land care facilitator, Lizzie asks, what are some of the most effective tools to take out to farm visits to demonstrate differences between conventional and regenerative, like your refractometers? And bearing in mind that cheaper options are good as we have no budgets. I'm sorry to hear that. Refractometers are very cheap. But yeah, I find some of the most effective tools is your infiltration rings. So looking at how well is water moving into the system, and the slaking test as well can be really dramatic. So slaking is where you put soil into water and look at how quickly does that disperse and fall apart, and it's biology that actually maintain that aggregate structure and stop it blowing. These big wind storms and dust storms that we're seeing in Australia at the moment, that is not natural, right? And what we're seeing on regenerative operations is the soil is not moving. And we've done some pretty good work on cropping operations where you can see that soil won't move if biology are working. So they'll hold that soil together. So slaking tests and infiltration tests, I think, are really powerful. Yeah, I think that's pretty good. We're doing a tea bag test, so you're burying tea bags in the ground in different types of tea and comparing that to conventional and regenerative, how quickly they break down. So they don't have to be expensive tests, but they're quite fun. There's some dye that you can put that in with your infiltration test, and dye that water as it moves through, and then cut like a trench so you can have a look at how is that water moving through, and look at the difference between hydrophobic conditions, because you guys have major hydrophobic issues, and regenerative practices should overcome those hydrophobic conditions pretty fast. Question from Sophia: what are some groups, businesses, institutions you would recommend to get connected to for someone that's starting an organic farming journey from scratch? Thinking about what to do when you finish your ag science degree. Good question. I would, yeah, loop in with the Regen Ag groups in Western Australia, SRF, so sustainable. Oh, I always mess it up. I even asked them again the other day. Anyway, they're a group of businesses that have got together that often put on workshops. I see Lismore University and yeah, there's a couple of universities in Australia now offering stuff, but I think just get connected to farmers, and there's some really, really great regenerative ag discussion groups on Facebook. So if you're on Facebook, get on that, and then look up hashtags for regenerative ag on Instagram and connect to those people, and yeah, just see who aligns with you and where you're going to be living. Can you feed sheep humate too? Yes, you can. I've only ever heard once of a story of humate going badly and people were drenching sheep and it was getting into their lungs. So we don't recommend drenching sheep. Yeah, so you absolutely, you can feed sheep, but we just leave it out free choice. We don't add anything to it. But I know there's some YouTube videos showing mixing biochar rather than humate with molasses in Australia and showing how the dung beetles just go crazy. So when I say soft brown coal, I don't mean biochar. They are distinctly different. Jono asks, are there more specifics of the humate product you're talking about? There's a specific product you found to be better than others? Yes, you don't want a humate to be too close to coal. Like, it shouldn't be black. It shouldn't be that black shale. It depends what you're wanting. So if you're just getting raw humate, you know, it's going to be you're going to be putting a lot more on. It's going to take a long time to release. But there are really good soluble products in Australia, and there's lots of companies that do it. It's interesting. There are humate seams in Australia, but I'm seeing a lot of them importing from here in, not far from where I am, here in Montana. Haley asks, hard claypan country, what's best way to promote plant growth? Yeah, you want to get curious, I guess, like, is that soil very tight because it's very high in magnesium? Is it a mineral imbalance? Like, I recommend actually you get hold of my book and have a look at what is it that's restricting growth. That's a really big, broad question. So I think everything that we've talked about, if you've got livestock, using them as tools. Do soil diseases like sclerotinia indicate a soil deficiency? And then how do you correct it? Well, yes, they do. What you find is a lot of those disorders can be linked to trace elements, particularly manganese. And so operations that have been using things like glyphosate for a long time have bound up the trace elements that the plants need to defend themselves. What we find is, as we address our microbial conditions, we don't see sclerotinia. So doing seed treatments, we might use trichoderma, we might use mycorrhizal fungi, bacillus, different types of organisms that we're putting down with the seed, and then we don't see sclerotinia. So it's like we're going to send out the army to defend it. But yes, there are trace elements involved in that. I would recommend you get hold of Don Huber's book on mineral nutrition of plants, I think it's called. Anyway, it's quite tricky to get hold of, but it's very good, and he covers sclerotinia in depth, as well as other diseases, and looking at what are the trace elements or the minerals involved in it. Quite a lot of you guys have issues with silica and silica function, and a lot of these fungal diseases relate to silica. So how do we get silica activated? And there are some quite good silica products that are now on the market that are also going to help you deal with your high aluminium. Some of the grain that's coming out of Western Australia is loaded with aluminium, and the silica products will help with that, you know. So are we growing food for humans? Cool. Thanks, Alan, for the nice comments. So does the book go into breaking down forest soils that are only growing stringy bark and yellow box trees to promote diversity? No, it doesn't really go into that, breaking down forest soils. It goes a little bit into forest soils, but not into the stringy bark yellow box. It's a fairly specific. What was Don's book called? I think if you just Google Don Huber mineral nutrition, I think it's, oh, anyway, yeah. It's in my cupboard, but I can't reach it from here. So can one add humic and or fulvic acids to fish emulsions, and at what rate? Talk to companies because different products have different concentrations. We're typically doing anything from 1 litre to 10 litres depending on what the product is, but a little bit is often all that you need. And yeah, it depends on the fish product as well, so you want a really good fish product. So Samantha says, will I be doing another talk? I'm doing a talk for Region WA on the 14th of May. I'll just I'll answer this one. People say we don't want weeds because they steal moisture in the soil. Is that research has really been refuted. You might get 1 year where that might be true, but a lot of what we're seeing in terms of weeds causing reductions in yields in the following crop is more because of an allelopathic or biological function. So it's not necessarily weeds. You want ground cover. You want ground cover because you want to be opening up the soil. I really recommend you guys have a look at Grant Sims' operation, who's Victoria No-Till, and see what he's doing in terms of having ground cover crops, and just how that moisture performs right through his whole profile, as opposed to you get no ground cover, so you create more bacterial soils, you start to have soil structure losses, more compaction, and now you find water doesn't move into the system. So there was work in Saskatchewan and Montana that showed that long-term chemical fallow actually had less moisture because you're burning up your carbon. So the whole system just starts to deteriorate. So we've really got to get over this focus on like weeds are bad. We just need to have some ground cover